Okay, so the last class we have seen different models for describing a impedance of a film, right. So, what I want to show today is few example applications, how the impedance spectra have been analyzed and used in the literature. So, I want to describe 4 examples, 2 of them from our group and 2 of them from research in other labs. First example is on corrosion, where basically we use electrically equivalent circuit. Okay. The second example is on CO carbon monoxide poisoning in fuel cells, okay, and that uses basically EEC. The third example is on biosensor, and that uses EEC, and the last example is from our group, which is using mechanistic analysis. Most of the literature, if you see, they would use equivalent circuit, because it is lot easier to work with, lot easier to fit a model for a given data. Okay. Interpretation, making sense out of it, sometimes it is easy, sometimes it is not so easy. In RMA, the analysis part is challenging. So, you do not see that many publications with mechanistic analysis. All right, the first example is measurement of or quantification of adsorption of an organic chemical called L-glutamic acid on copper electrode in nitric acid solutions. In nitric acid solution, copper will dissolve, but if the organic chemicals we put in, if it adsorbs on the surface, it can block or reduce the interaction between the nitric acid and copper. That basically acts as a corrosion inhibitor, although that was not necessarily the purpose for which it was used in the literature. So, what is done here is copper electrode in nitric acid at open circuit condition, open circuit potential, take impedance spectrum, and that is given by the figure that is given in the inset. You see, in the inset there are 2 plots. Okay. These are complex plane plots. The blue color plot, actually it shows a small semicircle and a 45-degree line. It is not very clear, the small semicircle is not very clear, but if you zoom in you would be able to see. That corresponds to 0 weight percent, that means no additive, no corrosion inhibitor. If you use a small quantity 0.05 weight percent of corrosion inhibitor, you see another semicircle in the green color, semicircle followed by the 45-degree line, and you can see the impedance values are larger, and both are in the inset. Third one is for little higher quantity, right, and that is the red color in the main figure C. Fourth one is 0.3 weight percentage, that is blue diamonds D, and the fifth one is 0.5 weight percentage, which is a blue circle, open circle. Sixth one is more or less same as the fifth one, the weight percentage is higher, but the impedance spectrum is similar to what you see for the fifth one. Okay. So, this is the spectrum that is obtained. Analysis is basically using following circuit. RS here is the resistance, RT is the charge transfer resistance, RS is the solution resistance, and instead of AA capacitor, it is modeled with a constant phase element, and Warburg element, because these experiments were done without rotation, stationary electrodes, solution was unstirred, and in addition you can also see it shows the 45-degree line at the low frequencies. So, when you fit this, I am not showing you the fits, it fits and you can get values. Solution resistance will be more or less the same, because small amount of this chemical is not going to change the solution resistance significantly. RT value will increase, meaning the semicircle diameter will keep increasing when I add more and more of this chemical. Okay. And there is a way to quantify that. Here I have shown you in weight percentage for this L-glutamic acid. The next slide actually we have calculated using concentration in molars or millimolars. Okay. After we get RT, we calculate a quantity called surface coverage. So, the way we visualize is this. Let us say this is the area of the electrode. If we say that we add a small quantity of the inhibitor, it can go and cover this surface. We add larger quantity, Of course, some of it will going to absorb, the adsorbed species will come out sometimes. So, it is going to be under steady state condition. If you have more concentration in the liquid, it is more likely the surface coverage is larger. When surface coverage is larger, we say that only the remaining area is available for the actual reaction. So, the way we define surface coverage here is, or way we extract it is, if we say that with the inhibitor we get a charge transfer resistance, without the inhibitor, that is at 0 weight percentage we get a charge transfer resistance, we can estimate the surface coverage using this formula. When RT inhibitor is same as RT, you will get 0 coverage. When RT inhibitor is very high, that means if the entire surface is covered with this inhibitor, charge transfer resistance will tend towards infinity. It is not going to be infinity, but it is going to be a large number, in which case we are going to say this number is much larger than RT, therefore, this will look approximately equal to 1. So, from the RT value we estimate the surface coverage value. From the RT value of this electrode with inhibitor and without inhibitor, we estimate this surface coverage. Because we expect this to be just a physical process of adsorption and blocking. So, it is effectively reducing the area of the electrode available for the reaction. So, that is how that reduces the net corrosion rate, or that is how we visualize this process. And if you plot the fractional surface coverage as a function of concentration in the liquid, okay, curve we get looks like this. At 0 concentration, of course, you will get 0 surface coverage, and it increases quickly and then it more or less saturates, which gives us the idea that it is probably described by Langmuir isotherm. Okay. Langmuir isotherm is described by this equation, where C is the concentration in liquid, theta is the surface coverage of the same species, and usually people will rearrange it and say this is going to be described by a linear equation, and it is easier to verify it. But then now you have enough number of software tools that you can use this actual equation and use nonlinear fit. That is actually a better description than getting this, because you want to minimize the error in theta experimental and theta model, you want to minimize this error. Here what it does is it minimizes the error of C by theta, and that means there is going to be a difference in the value of k that you get, there is going to be a difference in the confidence value of k that you get. So, just to say that just because previous literature shows you this formula and says they minimized based on this, it does not mean you will have to do it that way. Now that we have access to easy nonlinear fit tools, it is better to use the actual equation and fit it. And when you fit it, you would get a value for k, and that k is related to the del G value. 55.5 is actually the concentration of water in solution, basically it is pretty much water, and del G value you can get it using this formula. This is not the best way. If you really want to get it correctly, you will have to do it at various temperatures and then see if the k really follows this equation. If I vary the temperature, what happens here, but then what we got is with one value, one temperature. So, we basically said we can describe this adsorption. We wanted to show that this material adsorbs onto this surface. We were able to show by saying that the impedance value increases, and then we are also able to quantify a little better. Second example is in a fuel cell. Okay. In a fuel cell you have hydrogen, this is the overall reaction. In the hydrogen fuel cell, actually what you have is hydrogen splitting to form H plus on one side, oxygen not just oxygen directly reacting with hydrogen or H plus, it is going to combine with water and form OH ions. They will react with H plus ion and form water. So, on one side, anode side in this fuel cell, the anode side is where this happens. Okay. cathode side is where the oxygen reduction happens. On the anode side you have H plus, electron comes through the load, whatever load we give, and then comes to the cathode side where oxygen along with the electron and the water molecule forms OH minus, and the hydrogen formed on the anode side moves through this, combines with the OH minus and forms water. So, this will give you 2 H2 will give you 4 H plus and 4 electron minus. So, net reaction will still be 2 H2 plus O2, although water is necessary to actually conduct this reaction. Okay. And here hydrogen ion moves through this. There are other scenarios where OH minus will move through this. Alkaline fuel cell, it can happen where the movement occurs because of the OH minus ion. We are not going to worry about that. Hydrogen is produced usually from natural gas. Okay. So, it is from CH4, it is supposed to give you CO2 and H2, but a small amount of CO may sneak through. So, that is causing CO poisoning. Oxygen is actually just air, we do not supply oxygen, we usually supply air. Now, this study was done in the following manner. Okay. In an actual fuel cell, you will have oxygen along with moisture, controlled level going through one side, hydrogen coming through the other side with appropriate catalysts, and then you will have the energy produced or electrons coming out through this. Here what they have done is they sealed this, that means it is pressurized tube with hydrogen, but it is not flowing. Okay. Oxygen of course flows on this side. CO is not a good gas anyway. Okay. So, you do not want to pump it and release it, and it is lot easier to maintain a concentration if you are actually blocking it. So, what they have done is taken the spectrum before poisoning. So, that is actually a small semicircle here, it is not very clear, but it is a dark brown line indicated by this number 1. It is very small, so you cannot see it here in this scale. After giving small amount of CO poisoning, after an hour looks like this. After hour and a half, actually 2 semicircles are merged together, and in addition you have an inductive loop. So, initially it looks like this, second this loop becomes larger and larger, that it actually becomes like one semicircle. It is not a semicircle really, it is actually 2 semicircles combined, comes like this. You can see it is not really a semicircle, right. It is not even distorted semicircle, like it is not depressed semicircle where it is just pushed down. It is actually, you can visualize this as 2 semicircles merged together. One is a smaller semicircle, another is changing with time. Initially it was small, later it was larger, later it was even larger. Therefore, it looks like a distorted semicircle, and at even later time it goes like this. Okay. So, looking at this, you can definitely say mid and low frequency impedance increases over time. That means, the process of poisoning is continuing. It is a fixed concentration of CO, but it has not come to a steady state yet. Over time it keeps increasing. Okay. Of course, when CO poisoning happens in real cell, what will happen is you will not be able to produce that much current. The efficiency of the catalyst will go down. Okay. But this is just a study to understand whether you can get a signature out of this. Analysis is done with the following circuit. Okay. You have cathode to represent the oxygen reaction, they have represented that using a simple reaction, simple resistance and a constant phase element. In between cathode and anode, you have a membrane resistance, I am denoting that as solution resistance here, it is basically a simple resistor. And on the other side you have anode. Of course, when you have cathode, membrane, anode, and if you assume there are 3 distinct elements, it does not matter whether I mix it this way or whether I put it lined up like this. So, I have put cathode here, anode here, and then the membrane resistance. It is going to give me the same impedance anyway. I have actually copied from their diagram and just drawn it on my own, but I have followed the same sequence. Now, in the anode side, for the double layer it can be modeled using simple capacitor. This, whatever is shown in the red dashed box, okay, that is supposed to represent all the processes occurring there, Faradaic processes. So, that is denoted by 1 charge transfer resistance RT of the anode. Second, Warburg impedance, finite diffusion impedance. Okay. Third, they also use a pair of R and L, Maxwell element, but with inductor, and they denote that by something called relaxation impedance. Okay. So, at this stage I want to make some remarks. First, this system is modeled using a cathode with a resistor and a constant phase element. On the anode side, it is modeled with a Maxwell element and a finite Warburg impedance or bounded Warburg impedance is added to that. Okay. Ideally, what would we want to do? First of all, the oxygen reduction reaction, that is not a simple electron transfer reaction, it goes through multiple steps. So, it is a fairly complex reaction. On the anode side, we have two reactions, one is hydrogen oxidation and another is carbon monoxide oxidation. So, hydrogen oxidation reaction, that also does not happen with a single step, it happens with multiple steps. There are studies exclusively for these hydrogen oxidation reactions, and at least you need one adsorbed intermediate to describe that reaction. And carbon monoxide poisoning, okay, the way it occurs is carbon monoxide adsorbs onto the catalyst surface and then it gets oxidized very slowly. So, essentially it adsorbs and blocks the catalyst surface, and that is how it poisons, and that will need at least one adsorbed intermediate. So, we will need two adsorbed intermediates to describe the reactions in the anode side, and we will need one adsorbed intermediate, at least one adsorbed intermediate on the cathode side. So, it is actually complex multi-step reactions. This is just a pictorial of what one would expect to see. Carbon monoxide can bind possibly in one site, can possibly on two sites. These are called linear or bridge mechanisms, but this is how people have described this, and ideally we would want to use some mechanisms either proposed in the literature or something that one can propose ourselves, we can propose ourselves and use that to describe the system. Okay. Not just that, when you want to include mass transfer effects, and if you know that mass transfer effects are important, you cannot normally add a Warburg impedance to the Faradaic impedance that is obtained assuming mass transfer is rapid, because diffusion and kinetics are coupled via boundary conditions, and in the simplest case, that is simple electron transfer reaction, at open circuit potential one can write nicely separated expressions, one for the charge transfer kinetics and another for the mass transfer. But in general, we have not derived any expression for complex reactions with mass transfer limitations. We just mentioned that it is possible, but it is actually extremely difficult, okay. And it is not easy to separate, but right now what is done is that you have possibly one adsorbed intermediate that is described by the charge transfer resistance along with the Maxwell couple, that is denoted by Rk and Lk, and then mass transfer impedance is added as Zc or Zw, bounded Warburg impedance. The third point is, ideally I would like to conduct an experiment with 3 electrode system with one electrode as the reference electrode, and the potential should be measured with respect to the reference electrode. But then because of challenges involved in conducting such experiments in a fuel cell, 2 electrode system is used, which means it is somewhat difficult to tell what is the potential drop across each electrode, what is the potential drop across anode and what is the potential drop across cathode. Still, the authors have modeled in this fashion mainly because it is not that easy to even develop an expression for complex reactions even on only one side, even on the anode side with mass transfer limitation, and when you realize that you have to add the reactions on the cathode side also to calculate the total impedance, it becomes next to impossible. Even as of today in 2018, if you develop the expression, it might be possible for us to generate the spectrum for a given kinetic parameters and diffusivity parameter, but it is practically impossible to run it through an optimization program and fit and obtain the parameters. So, to make sense out of the data and to get an understanding, I would guess the authors have described this reaction in the anode side with one adsorbed intermediate species as a simplification, and then added the bounded Warburg impedance, added the diffusivity part to this, and that is how they have modeled this data.
How do you know you are supposed to introduce a Warburg element in this case?
Basically, if you say that the mass transfer effect is playing a role, you have to you can't introduce a Warburg element, that is my argument here, but you have to account for mass transfer by some means.
But you see the EI spectrum and then you displace it.
So, EI spectrum is one. Second, if you think the diffusion of the species is going to play a role, because if you put a thicker membrane, it is going to make an impact. You put a thicker catalyst layer or gas diffusion layer, it is going to make an impact, then mass transfer is playing a role. So, you can't just look at the spectrum all the time and say this is what is happening. I can look at the spectrum here and I can say it is probably Warburg impedance, but that is also because I know the solution is not stirred and the electrode is kept stationary. This type of data can come from another system. You may have a film in which diffusion can happen, then I may or may not be able to use this semi infinite Warburg impedance there. So, you have to know something about the system. Here they have modeled this, they have not shown the model fit, they have not tabulated the complete model result. What they have done is different duration, what is the value of RT, is it increasing, decreasing, and then they have interpreted it saying that okay, poisoning causes. The data is good, what happens when you have poisoning is clear from that. Whether this is the best model is arguable, and describing that you need an inductor and a resistor and saying that this describes a relaxation is something that you would see many times. So, whenever they are modeling something, you see an inductor, they would say it describes an relaxation of adsorbed species. So, I am not sure that is the best way to describe. Relaxation of adsorbed species could possibly mean they are interpreting it as one form of d theta by dE, but then d theta by dE can give you a capacitive behavior, can give you an inductive behavior. Capacitive meaning whatever spectra you get can come from another system which contains electrical resistance and a capacitor. It does not mean this variation of surface coverage with potential acts as a capacitor in the sense that it stores charges for you. Just like if it acts as an inductor, it does not mean there is a behavior like a coil with magnetic field. It just means whatever spectra you get here can be mimicked by another electrical system. Okay. I just wanted to show you that this is an example of interpretation of data. Third, give you a little bit of background. In biosensor, you want to detect presence of something called antigen. Antigen could be a parasite, malaria parasite, could be a dengue virus, okay, or some chemical that is specific to dengue virus. That virus may release a chemical. If you detect that chemical, you can say yes, this person, if you detect that chemical in the blood sample of a person, you can say this person is affected by dengue. You can either detect the virus or the chemical that is associated with the virus, released by virus, but it is marked, like if you see this chemical in the blood, this person has been exposed to this virus or this bacteria. Okay. The bacteria is called antigen, that chemical is also called as antigen. Okay. Whatever our body produces in response to that is called antibody. Okay. So, biosensor here is something where you want to detect the antigen. So, what you want to have is an antibody which is specific to this antigen. Now, we want to prepare an electrode. On top of the electrode, you attach or somehow suitably couple an antibody, cover the surface with antibody, After that you expose a blood stream, the blood stream may or may not contain the antigen. If the person is not infected, it should show nothing. If a person is infected, that antigen will bind to this antibody. Now it should show an increase in impedance, at least that is the expectation. So, to bind an antibody to an electrode, you have to go through certain steps. Gold electrode is normally used, although there are many other combinations available now, but it is kind of a starting point. Organic chemicals, right, alcohols and acid which contain sulfur, it is called mercaptan. These chemicals are first used to form what is called self assembled monolayer. Sulfur has an affinity to gold. So, if you put these chemicals, they will all line up in this fashion, or that is again the expectation. So, if I have an area, I put these chemicals, most of the area will be blocked and these chemicals will line up like this. So, it is called, it is assembled on their own, we are not doing anything to specifically assemble in a particular fashion, self assemble, and it forms a monolayer, one layer. Of course, it is not 100% true, it may assemble in different orientation and it may not assemble in some areas, those are there, but if it works as we expect, it is going to form a SAM. Then the SAM is not, the reason we are doing all this is because the antibody is not going to just sit on the surface well. So, we want to make the antibody sit on the surface, means we need to put a glue, and it does not work with one glue, we need to do one chemical, that is MUA-MH. In addition, we put two other chemicals, you expose the gold to this for certain number of hours, 12 hours, 18 hours, 24 hours. It forms a monolayer and then we expose to these chemicals for a couple of hours. So, it is not one layer glue, it is a multi-layer glue. Antibody can be coupled to this using these chemicals. So, these chemicals form a bond and then, so first this chemical forms a bond, then this chemical forms a different structure, and then if you put the antibody, it is going to sit on top. If you expose the antibody to the gold, it is not necessarily going to stick well. Not only that, antibody is a large molecule, it will have many orientations. Ideally, you want it to be exposed such that when the antigen comes, it is able to bind. If that is looking like this, when the antigen comes, it will not bind. So, there are difficulties, but right now the expectation is antibody will be attached to this with antigen coming and sitting on top of that. If the solution has antigen, so you have to build the electrode like this and then keep it ready. When somebody gives a blood sample, you will put a drop of that here, measure the impedance, and if it is high, you will say, depending on how high it is, you will say you are exposed to this virus or this bacteria, and level is serious, moderate, initial level, whatever that is.
So these long chains are the glue rate, like OH and OH.
See, S is binding to the gold, we need S here. You need long chains here. By the way, again I am not showing you really per scale, it is not the antibody is going to be small, antibody is going to be actually very large, but then it is going to go out of the slide. So, do not think it is done to scale, it is just a way to see. So, this is the idea, and after you expose, after you attach the antibody to this electrode, we normally saturate with one more biochemical called BSA, bovine serum albumin, that has a good affinity to gold. So, wherever the SAM is not formed, if certain area is not exposed to, it is exposed to SAM, but it is not attaching, BSA will go and block that area. So, that the antigen does not come here, sit here, antigen or any other chemical in the blood should not come here and sit here and increase the impedance. So, before exposing to blood, after putting the antibody, we initially feel that okay, SAM should block every area, and then on top of which we put EDCNHS, on top of which you put the antibody. Hopefully, the entire surface is filled and antibody is sitting outside waiting for antigen to come and capture that. We can't be sure, there might be vacant areas, so fill it with BSA. BSA will attach to gold, but not to this antibodies. Again that is the expectation, and when you actually see, it is not that easy. At least that is what we are seeing in our lab. We have to do it in a very, very controlled fashion, otherwise it is not easy to get repeatable results. Coming here, this is a published data. So, bare electrode is not shown. When you put SAM layer, impedance is in the range of 200 kilo ohm centimeter square in the real value, imaginary value is around 50. So, you can see it is a large value. When you put NHS, it actually decreases. What that means is, it is not completely blocking. When you put the NHS, it allows the reaction to happen. This reaction is usually Fe2 to Fe3, ferroferry. Okay. So, that means open areas will allow some reaction to happen. Even these areas with SAM, they will allow the reaction to happen at a much slower rate. When I put SAM and NHS, it allows the reaction to happen at a faster rate, little faster rate. So, that is why the impedance comes down. And then I attach the antibody, I do not attach the antibody, the group has attached the antibody and impedance has increased. When you expose to BSA and you block the remaining area, you can again see it is increasing. That means there were some vacant areas where BSA is blocking. Now, this electrode is ready for testing. This electrode, when you expose it to solution which does not contain the antigen, and this is for a particular bacteria, I suppose, Listeria, some such bacteria, I do not remember the name now. When you expose to different quantities, it is expressed in what is called colony forming units per ml. You can think of it as concentration. You have higher concentration here, impedance actually increases. And this is modeled using the circuit, typical circuit of solution resistance, AA represented by CPE, charge transfer resistance and Warburg. And what they show is a plot of charge transfer resistance with respect to colony forming units. Again, it forms something like a Langmuir isotherm, although it has not really saturated yet. What people ideally want is a linear regime, where higher concentration gives you proportionately more signal, but what they got is a nonlinear one. But yet, if you take an unknown solution, if the charge transfer resistance comes out to be in 375 kilo ohms centimeter square, you would say this likely to have a Listeria of this much concentration. Okay. And of course, this is also tested with other type of bacteria, and they say that it does not offer any significant increase. That means, the antibody is specific, it is not binding to any other bacteria. The reason you are doing this is because what if some surface is still exposed and this antigen is coming and binding, maybe some other chemical in this solution is coming and binding. So, you normally will test it with other type of material that is likely to be present. Okay. That is looking for specificity. Is it specific to this entity, this antigen, or is it going to be blocked by anything? Okay. And the last one is data from our group. Polarization, it shows an increase in current and then decrease. It offers a passivation. Okay. This is niobium, which is the metal like tantalum. It does not dissolve in acid except in hydrofluoric acid. When I say it does not dissolve, I mean if you take very concentrated sulfuric acid, it will probably dissolve in ppb level, but not to a level where you think of it as a corrosion in a short term or even in medium term, it is not going to be a big problem. HF, it will etch at a significant rate. Okay. If you increase the potential, niobium will form niobium oxide. At normal atmosphere, it will form a very thin oxide. If you go to higher potential, especially if you expose it in solution with sulfuric acid, any of this where you can have water or oxygen present, it will form a thicker oxide and form what is called passivation. Even in HF, if you go to large enough potential, I will form a thick oxide, depends on what is the concentration of HF. If you have a small concentration, very quickly it will form a thick oxide. If you have a large concentration, because it dissolves the oxide fast, I will have to go to very high potential to get a thicker oxide. Now, we can get impedance spectra. We have taken it at 3 different potential in the region where current increases. and then 3 different potential in the region where current decreases. So, it forms 2 semicircles, and this is in rotating electrode. So, if you increase the rotation beyond certain limit, mass transfer is not a problem. So, we say it is all kinetic limited. 2 semicircle, but then the semicircle diameter increases. This first semicircle diameter decreases, it goes from close to 18 to 9 to even less than that. Second semicircle diameter increases, right, it ends at around 25, not really increases, it is probably similar here. This ends up being in the larger side, and there is a small loop here. So, this first and second were modeled using 1 and 2. Third, if I want to model, no, this first and second were modeled using only one RC element. I will get one semicircle from here, so that is a high frequency loop. I will get one semicircle from here, that is the low frequency loop. Here again 2 semicircles are sufficient. I mean 2 RC elements are sufficient, one Maxwell element is sufficient. Here to model this inductive loop, I have to use either an inductor or I have to use an RC with a negative value. You could get data in the passive region, you get something which is mid frequency inductive loop, which comes clear at higher voltages, but it is very small, unless you zoom in you will not be able to see at the lower potentials. Again you can model this with the same circuit, but then getting physical interpretation is a lot harder. So, what we have done is to use mechanistic analysis. Fit is not as good. First of all, in EEC you do not even predict the value of polarization current, that means I am not predicting how the current will vary. I just model the impedance spectrum, one spectrum at a time. Here we predicted the current, we predicted the impedance spectrum, the match is, you can say qualitative, meaning whatever main feature you see, you can see here, maybe semi quantitative to say it is somewhat similar to this. It is definitely not as good as what you would see in equivalent circuit analysis. However, just to show that okay, you can predict the feature, although not the data as well. However, what we get out of this is the following. We propose a mechanism like this: niobium gets oxidized to suboxide NBO, which gets oxidized to what is called pentoxide. This is the most stable form, but this is also possible. And niobium can get oxidized to suboxide, it can get oxidized to pentoxide, both are present on the surface, and the pentoxide can dissolve, that is one pathway. But that pathway will not give me an impedance spectrum like this, it will not give me a current potential diagram like this. So, I will have to propose another reaction, which is the suboxide goes into solution while losing 3 electrons, that is Nb2 plus goes into solution, not 2 electrons, while losing 3 electrons. If I do not have this step, I will definitely not get a curve like this for current potential diagram. I will definitely not be able to get all the features in impedance spectrum. I might be able to get one spectrum in the active region, that I can fit well. Even in this model, right, if I fit only 1 or 2 spectra, I can fit lot better than what I have shown here. But we want to simultaneously fit all the spectrum, that means fit may not be as good, but this is a better description where one model fits all the data at multiple potentials, it also predicts the polarization data correctly. This is better than using a smaller set of data or one impedance spectrum, and this gives an idea using this model, saying okay, this model seems to describe this phenomena reasonably well. Let us see what it predicts. When I go to low over potential, this curve, when I go to low over potential, it is mostly bare niobium. Any suboxide and pentoxide that is formed is taken into solution. Because with this, once I know the rate constants, because of time and space, I am not showing you all the rate constants in tabulated form, but you have the values of rate constants, you can predict what will be the surface coverage of theta 1, theta 2 as a function of potential. When you go to mid potential, what you see is some niobium oxide is present as pentoxide, some is present as suboxide NBO, and some vacant surface is also present. Even though they are simultaneously dissolving, some of the niobium is getting oxidized to NBO, some of the NBO is getting oxidized to Nb2O5, some of it is going into solution, some of the oxide is also going into solution, but the oxide formation rate is more, suboxide to pentoxide is more. Therefore, under steady state conditions surface coverage is more for both of these. When we go to very high potential, what happens is pentoxide becomes more or less covers the entire surface. There is still some niobium left, there is still some suboxide left under steady state condition. Left meaning I do not mean to say that pentoxide remains there, some suboxide remains and niobium remains. What I mean to say is net, if I look at it now versus if I look at it later, surface coverage, that means some pentoxide might have gone into solution, this might have become niobium now, because underlying metal is exposed. Some of the suboxide might have gone into solution and exposed this as underlying material. Some of the suboxide might have converted to pentoxide, that means it is not moved out, but the color has changed, color here is just a representation, right. Some of the niobium would have converted to suboxide, but net if you look at it now versus few seconds later, on the average I will say maybe 95% will be pentoxide, 3% may be suboxide and 2% may be niobium. I do not mean to say the locations remain the same, I mean to say the surface coverage when I look at it on the average is going to remain the same under steady state condition. And this gives you, I think this gives you a better idea of what is happening compared to saying these are the element values here, although fit is better in that situation. Okay, we will stop here now.